Hello, everyone, and welcome to today's live broadcast, Multiplex Immunofluorescence, Profiling of Tumor Biopsies using UltraPlex mxIF Technology, presented by Dr. Marie-Louise Loupart, Image Analysis Specialist for Leica Biosystems, Aperio ePathology, and Dr. Helen Snyder, Director of Preclinical Development and Strategic Partnerships at CellIDX. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Leica Biosystems. For more information on our sponsors, please visit leicaBiosystems.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble hearing or seeing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. We have two presenters for you today. Speaking first is Dr. Helen Snyder. Dr. Snyder received her PhD in immunology from the University of Cambridge, UK, and trained as a postdoctoral fellow at Scripps Research Institute, La Jolla, California. Dr. Snyder has over 30 years experience in immunology research, reagent development, preclinical development of biotherapeutics, and business development of immunoassay and immunodiagnostic products. Following Dr. Snyder is Dr. Marie-Louise Loupart. Dr. Loupart has 13 years of experience in biomedical research, being trained as a molecular biologist, extending into cell biology, cytogenetics, developmental biology, and biochemistry during the course of her research career. For complete biographies on our speakers, please click on the Speaker Biographies tab above the slide window. Dr. Snyder, you may begin our presentation.
Thank you for joining us today for this joint CellIDX Leica presentation on multiplex immunofluorescence profiling with tumor biopsies using UltraPlex mxIF technology. We see this as the final piece of the puzzle in analyzing tumor biopsies. Much information has been gained from genomic analysis as well as multi-parametric analysis of dispersed tumor samples by flow cytometry. While this provides valuable information on what cell subsets are present, particularly immune subsets, which are only defined by multiple markers, as well as their level of regulatory molecules such as PD-1 or PD-L1. What has been missing is context. In other words, where each of those subsets is located relative to each other and to the tumor. Information obviously lacking once the tissue architecture has been disrupted. The UltraPlex technology was developed to meet this unmet need for a technology which is robust, quantitative, and multiplexed, capable of detecting multiple biomarkers in a single tissue section, and which is fully compatible with current pathology lab workflows. Here are the goals we had in mind when we developed the technology. We wanted a technology that was sensitive and specific, that was fast and simple. Basically, what we were aiming for was a technology which followed a simple, standard path lab protocol of 2 steps and 2 hours. We also wanted one that was practical and robust. Current approaches to multiplex include limited 2 to 3 marker staining by selecting primary antibodies of different species or isotypes, followed by staining using anti-species specific secondaries. This is very limited, particularly as most antibodies that are used are either mouse or rabbit, increasingly rabbit, and that only has 1 isotype. Or alternative approaches to multiplexing use repeated rounds of stripping and staining, which is both lengthy and also requires very careful planning of the order in which the markers are stained for each panel. So what we wanted was a much more practical and robust approach. We wanted one that was reproducible and quantitative, and one that was scalable and easily adoptable. Again, we were aiming for a kind of plug-and-play technology which could be widely used. We also wanted one that conserves tissue, given that we have now progressed beyond simply looking at diagnostic biopsies to frequently looking at multiple in-treatment biopsies, which are usually very tiny and also precious. Those tiny biopsies are divided up into analysis by multiple different technologies. So obviously we wanted to get as much information as we could from a single small tissue section, maybe one or two. We also wanted a technology that was broadly applicable to analysis of all tissues. So we frequently describe our technology as flow cytometry in situ. And so people often ask why we can't just use the same directly labeled primary antibodies that we use in flow cytometry. Well, occasionally you can, but as you can see here, what we have is staining of a breast tumor biopsy with antibodies against HER2 in red, the estrogen receptor in blue, and the progesterone receptor in green. As you can see, the intensity of staining is really pretty poor on this directly labeled specimen on the left. However, on the right, you can see the amplification of fluorescence intensity you get when you use a secondary antibody amplification. So here we have we have an unlabeled primary detected using anti-species specific secondaries. And you can see the very nice crisp membrane staining for HER2 in red. However, this is limited, obviously, as I mentioned before, because there are not very many different species of antibodies, and so this really isn't amenable to multiplexing. So here's what you get when you use our UltraPlex technology. You can see very beautiful crisp membrane staining in red for HER2 and nuclear staining in blue for ER and green for PR. So we get about 4 to 5 times the signal you get with directly labeled primary antibodies, and we get between 70 to about 120% of what we would see if we used a fluorescent labeled anti-species antibody. So it combines the ability to multiplex, but also amplify as well. So how does UltraPlex work? In UltraPlex, we add an individual different peptide barcode as a hapten-like tag to each primary antibody. So each primary gets a different peptide tag. This means that we can add all the different primary antibodies together as a cocktail in one step on the tissue section and incubate just for 1 hour. Then we wash and we come back with individual anti-peptide or anti-hapten secondaries, each of which is labeled with a different fluor. So it's simple: cocktail of primary, incubate, wash; cocktail of secondary, incubate, wash, and then you're done. A very simple technology. We routinely make standard 4-plex panels, but we're continually expanding our library of peptide tags, so we can also make 5 or 6-plex panels. The beauty of UltraPlex is that it doesn't matter whether we're looking at 3, 4, or 5 markers. It still takes the same 2 steps and 2 hours. So here is an example again of staining a luminal B breast cancer biopsy for HER2. You can see the nice membrane staining in red, the estrogen receptor, that's the nuclear staining in blue, and the progesterone receptor, the nuclear staining in green. And the 4th marker is a proliferation marker, Ki67, which is actually pink, but when it gets overlaid with the other nuclear markers, here it appears as that kind of orange color. So this highlights some key advantage of our technology. So the technology is primary antibody species independent. Of these 4 markers, HER2, ER, and PR are all rabbit monoclonals. Ki67 is a mouse monoclonal. So we are primary antibody species independent, agnostic. It doesn't matter to us. We can also detect multiple markers which co-express in the same cell compartment. So for example, ER, PR, and Ki67 are all nuclear markers. We can detect those very easily simultaneously. You can see here we get very good batch-to-batch reproducibility. We also have reproducibility, here we have serial slides stained with the same panel on the same day. So you get really good slide-to-slide reproducibility. We also get good batch-to-batch reproducibility and also day-to-day staining. And we currently have over 6 months stability on a number of our panels. So serial section overlay of 2, 3, or 4 sections stained with different 4-plex panels generates vast amounts of data. So the panels are modular. We can stain the different sections with a different panel. We can do image registration and overlay of those sections, and we call this superplexing. So up on the top left, there again is staining a breast cancer tumor with the breast cancer panel. Down there on the bottom right is a basic immune panel looking at CD20 cells, which are B cells, and CD3, CD4, and CD8 T cells. Actually, almost before I overlay these two, you can really tell what's going on, that the immune cells in this particular tumor are not gaining access to the tumor tissue. They are really very restricted here to the stroma. So as this be immediately apparent, one of the main applications for this technology is currently in the area of immuno-oncology, where it allows characterization of tumor-infiltrating immune cells, their phenotype and numbers, their levels of expression of checkpoint markers such as PD-1 and PD-L1, and localization of immune cells relative to the tumor and to each other. This potentially provides predictive markers of response in immunotherapy. The ability to identify multiple co-expressors allows detection of rare subpopulations of cells. So here you can see detection of, this is a basic immune panel staining tonsil. Here you can see co-expression of CD3 and CD4 on T helper cells and CD3 and CD8 on cytotoxic T cells. We can potentially look at multiple markers on each subset, so we can identify very rare subpopulations of cells. So down here at the bottom, you can see co-expression of CD4 and TBET, which identifies Th1 helper cells. CD4 and FOXP3 is the middle of the small panels at the bottom. Those would be Treg cells. Then we have CD8 and TBET expression. And then the panel over to the right is co-expression of CD4, TBET, and FOXP3, which is a rare subpopulation, which is actually a kind of super suppressor cell. So we stain multiple different tumor types. This slide shows staining of a head and neck squamous cell carcinoma with a panel looking at, on the upper left, PD-L1 expression on different macrophage subsets is shown by co-expression with either CD68 and CD163. That's the pink staining and the orange staining, which you can see in this sample is pretty much restricted to, the macrophages are pretty much restricted to the tumor. As opposed to PD-L1 expression on the tumor cells. So here the tumor cells are shown by the purple and the PD-L1 is the kind of aqua staining. So in this particular patient, most of the PD-L1 expression is on the tumor and most of the macrophages are restricted to the stroma. The bottom left panel is a T-cell subset panel. So you can see here very nicely, I'm not sure how it magnifies on your screen, but the FOXP3 staining is the red nucleus surrounded by the green membrane staining. So all those little red dots surrounded by green, those are all Treg cells. The yellow staining is looking at CD8 positive cytotoxic T cells. So when you do image registration and serial section overlay, you can see that you do get some infiltration of Tregs within the tumor, but most of the immune cells on the APCs are within the stroma. So I've only shown you 2 overlays here. I mean, obviously we can overlay 3 or 4, but it becomes very confusing to the eye. Really visually, we can just look at 2, but it's obviously very amenable to large data processing. This is another example showing staining of lung cancer cells with our PD-L1 panel. Unfortunately, I don't have a mouse, so I can't show this to you, but the tumor again is in red, and then if you look in the central area of the section, you can see kind of a golden orange staining. That is an overlay showing PD-L1 expression basically on the tumor. In this sample, there is very little PD-L1 on the macrophages, but those are shown very nicely by the kind of the purple center surrounded by the green staining. Those would be the CD68, CD163 double positive macrophages. And then if you kind of scan around the section, you can see some cells which are just purple. Those are just the CD68 single positive macrophages. This is another lung cancer tissue section, and you can see it's a highly infiltrated tumor. The CD20 positive B cells are shown in purple. There are very few green CD8 positive cells, but there definitely are some. And most of the T cells are the CD4 positive T helper cells. But here is a very intense infiltration in this particular tumor sample. This is staining of a thyroid gland papillary carcinoma. Again, very strong staining for PD-L1, that's the purple, restricted mainly to the tumor shown by pan-CK. But as you look around, there is a little bit of purple showing on the CD68, CD163 positive macrophages as well. Here, this is a T cell subset panel looking on staining of thyroid gland papillary carcinoma. And again, you can see the very nice FOXP3 staining, those red nuclei surrounded by green membrane staining. So those are Tregs. Cytotoxic T cells are in kind of aqua, and then PD-L1 staining is in the kind of pink color there. So these are our qualified off-the-shelf multiplex panels. However, a lot of what we do is custom or semi-custom panel development of 2 to 6 markers. Sometimes people want entirely new panels, or often they might want to switch one marker in a panel for another. You know, maybe they want to switch out PD-L1 for a different checkpoint marker. So we have additional markers available, some of which are TBET, LAG3, and granzyme B. We're also continually qualifying new antibodies and have multiple targets in development. Automated quantitative scoring, or AQS, was developed using Aperio's cellular immunofluorescence algorithm. So clinically, HER2 levels on a tumor are designated as 1+, 2+, or 3+, and on the basis of that, they decide whether to treat the patient with Herceptin or not. So here we show use of the algorithm to setting arbitrary cut points for HER2. And you can see down here on the bottom 2 panels, the blue cells are the 1+ cells, the green are the 2+ cells, and the pink are the 3+ cells. However, we're currently involved in a study where we're comparing HER2 staining using an UltraPlex panel with pathology scoring for HER2, so that in future we can set clinically relevant cut points for HER2. However, the main reason I mention this is because you can also apply this to looking at levels of expression for any marker. So here I'm showing PD-L1 expression. And as we all know at this point, while simply looking for numbers of PD-L1 expressing cells infiltrating a tumor doesn't give a reliable predictor of the response to PD-L1 checkpoint blockade, maybe also looking at levels of expression of PD-L1 combined with perhaps the presence of other cell types within the tumor might allow development of a predictive algorithm for who might respond to PD-L1 checkpoint inhibition or to other checkpoint inhibitors. So I'm sure that Marie-Louise will discuss this in more detail. But here we show combining UltraPlex analysis using staining with 2, 3, 4, 5-plex panels with use of the Aperio Genie pattern recognition software to identify tumor versus stromal regions. One of the lower panels there, you can see identification of the stroma in the kind of green color versus the tumor in pink. And then also combining that with the Aperio cellular immunofluorescence algorithm, which enables you to identify 8 different subsets based on whether they express what combination of PR, PR, and HER2 the different cell populations express. And you can combine all of those together to create a virtual map. So by combining multiplex staining with image registration of serial sections overlaid, we can look at 4, 8, 12, 16, or even 20-plex staining. Or if we use a 6-plex panel, we can look at 6, 12, 8-plex staining. That means that we can identify even very rare subpopulations of cells expressing multiple markers. We can look at the percentage of these cells as well as the intensity of staining. We can calculate phenotype ratios and perform proximity measurements of subsets of cells relative not only to each other, as well as their presence within the stroma versus the tumor region and distance to the tumor margins. So this generates big data, obviously very amenable to machine learning and artificial intelligence. So in summary, UltraPlex allows simultaneous detection of multiple markers on a single tissue section, high throughput, simple and rapid staining using cocktails of antibodies and a single antigen retrieval step. It is primary antibody species independent and compatible with autostainers, images, and image analysis software. So through our CRO division, PathIDX, we provide both custom panel development. A lot of what we have are obviously immuno-oncology panels, immune panels, and breast cancer panels, but we can generate any custom panel that's required. We offer staining services, imaging services, and also image analysis services. So we welcome everyone to visit the website, and please don't hesitate to contact me at any time if you have any questions. And I think at this point, I'll just pass it over to Marie-Louise.
Hello, everybody. Thank you for joining us today. Now I'm going to talk to you about the Leica solution for fluorescence multiplex imaging. The products I'm going to discuss are intended for research use only. They're not intended for diagnostic purposes. Today, multiplex is most commonly visualized with fluorescence using technologies such as the elegant UltraPlex that Dr. Snyder has just shown us. Multiplex assays benefit hugely from automation at every step. Stain, scan, manage, and analyze. And these are the 4 topics of my part of the presentation. Our products are designed to work on both bright field and fluorescent slides, 1-plex, 2-plex, or many-plex assays, special stains, biomarkers such as proteins and nucleic acids all form assays that we can actually manage on our stain, scan, manage, and analyze systems. So from stain through scan, manage, and analyze, our platforms are designed to work together or complement any manual steps the user prefers to rely on. So here are some examples of different staining protocols performed on the Bond RX. I'm going to concentrate on the 2 lower images. They're fluorescent and they're clearly multiplex. The lower left has 6 colors. That's actually a 5-plex assay with a DAPI counter stain. The lower right image is a 4-plex assay, again using DAPI as a counter stain. These are typical of immuno-oncology multiplex assays that are in vogue today. Basically, these sorts of assays are all about phenotyping the cells. The immune cells interacting with the tumor can be distinguished by specific biomarkers, such as helper T cells are stained with CD4, killer T cells are stained with CD8, B cells are marked up with CD20, regulated T cells are marked up with FOXP3, and the tumor cells are usually denoted with something like cytokeratin. What's critical about this is it's rarely that the stains overlap unless the cells are overlapping in a very crowded, highly dense tissue structure. But it's usually about identifying individual cells that have 1 or maybe 2 stains associated with them. So how do we get to these images? Let's look at my first topic, which is about the autostainers. So there are currently 2 machines in the RX range of automatic stainers, and these are intended for research. The freestanding RX there on the left, and then its baby brother, the benchtop RXM. They're basically both going to do the same job for you, performing fully automated stain protocols for up to 30 slides in 3 trays. The main difference is the size of the reagent tanks and the dump tanks. Bond is easy to use. It's fast, consistent, and will get your slides done with a minimum fuss. The 3 independent trays mean that up to 3 different protocols can be carried out simultaneously. So you have full flexibility there. Even better, bonds may have been pre-programmed with protocols to work specifically with Leica Biosystems reagents. More importantly, it's actually open for other detection kits and reagents, so you don't have to abandon your favorite system in the lab just because you're taking on an automatic stainer, such as the Bond. Bond also supports custom protocols. What I mean by this is the user can set the number, the order, the time, even the temperature of every step. So you have full control here. Once the staining is complete, your slides, maybe 1 slide, maybe 30 slides, or however many you're running through your system, these then enter what I call the Aperio zone. And this is the second part of my talk. It's the second topic. After staining, the user could visualize the slide through a standard fluorescent microscope. All I can say is good luck to that. I've done that kind of thing for years, and it's horrible. Multiplex images often utilize fluors in the near-infrared range, which are not visible to the human eye. So you need to actually digitize multiplex slides to start with. Even if you're not using Sci5 or one of these other practically invisible markers that you can't see with the naked eye, it's still incredibly difficult to get it straight in your mind, all the different stains that you may be looking at. So the Aperio Versus scanner is a research scanner that's designed to handle multiplex slides with ease, really take all the pain away, just like the Bond can. It will generate perfectly aligned, tile-based fluorescent images in the range from 350 nanometers up to 750 nanometers. To you and me, that's anything from DAPI to size 7 included. That's a pretty broad spectrum. You get to choose between the 8-bay or the 200 loader, and it's the loader that you can actually see there. And it's up to you when and how you those 30 slides that will come off the Bond. Versa also has an 8-position dichroic turret buried in the heart of the microscope. It handles all sorts of different fluors for your multiplex assay with ease, using a very standard fluorescence system. Versa has a range of objectives to choose from too. Fluorescent slides can be scanned at any magnification from 5x to 63x. So you're not relying on a magnified 20x image, but true resolution at 40 and 63x, using 40 and 63x objectives. Do you want even more? Well, you can actually apply an automatic oiler to the system and use oil immersion lenses for 40 and 63. And then these really enable you to use the scanner for fish scanning very easily. It's great for tissue fish. So this is what you get. Beautiful images from a highly flexible scanner with a simple user interface. Look at setup and scanning. It's got all 200 slides there. All you have to do is set your scan template in the top right, click on the file destination in the bottom left, and then highlight the slides for that scan template. If you want to have different destinations and scan templates in your 200 batch or even in your 8 batch if you're using the 8-bay, you can. You can mix bright filtering fluorescence in the same batch. This system doesn't care. It will just take the slide, knows what it's got to do with it, and do it for you perfectly. You want to see what's happening at every pass, at the end of every pass? Well, you can do. You can review and export the scans manually, or you can just believe it, let it get on with its job, and scan the whole lot for you. You can monitor it easily. At a glance, you can see that 7 of those slides are ready for review. 1 of them is in red. That's telling me that it's not actually been happy with what it's tried to scan. You have the chance to go back, review it, tell it to scan it again, or you can dump it and continue with the other 7 as you like. Calibration can be as automatic or as flexible and in-depth as you want. For fluorescence, you can set automatic exposure for every fluor. Instead, you can actually set a variety of different settings for each fluor. You can have some on automatic, some on manual exposure that you calibrate yourself on your slides. You can also set Z stacks, and they can be different for every fluor. So you've got full control here. So with Versa, you can now capture as much detail for as many cells as you want on your multiplex slides. Why does magnification matter when looking at fluorescent images? Well, the obvious answer is that it does. But let's go through a little bit of why and what we do to overcome some of the limitations. Low resolution is great. You get a lot of cells in one single tile, or what we should really call field of view. You've got good focus, but unfortunately the detail is not great. Two small spots stuck together are going to be very hard to see clearly at low magnification. But you've got good focus. To get more detail, you need to increase resolution. Use a higher magnification objective. But you get a shorter focal depth. You also, really annoyingly, get fewer cells in your field of view. So you end up having to take more tiles, capture more fields of view to get the same number of cells as at low power. The focus becomes more critical with higher magnification objectives. And it's very important that you handle this properly. Here at Leica Biosystems, we actually solve this with a time-honored fashion within our company, and that's using Z stacking. So what we do is that we take Z stacks going above, through, and below the perceived focal points, focal plane for each floor to ensure your tissue is always perfectly captured. Managing these Z stacks, the system then figures out what signal is actually in focus and what is hazy and unfocused using something called nearest neighbor deconvolution methodology. Not a physicist or a mathematician, but all I can say is, in my opinion, it works brilliantly. Look at those two images in the bottom right. You generate a maximum intensity projection, which is a summation of all those signals that are in focus on different Z stacks. But you don't, in our case, we actually keep the maximum intensity projection and also the input Z stacks. You can always go back to those raw data and step through. But the level of detail you now get because of this deconvolution is outstanding. And you can now interpret your multiplex slides with ease and a great deal of confidence. The Biosciences generates whole slide images at any magnification. We say it scans from 5x to 63x, and you can do a whole slide at any of those magnifications in between 5x, 10, 20, 40, or 63. You're a little bit crazy to consider doing multiplex and 63 with Z stacks for 7 or 8 colors on the whole slide. That's going to be several tens, if not hundreds of gigabytes in size, but hey, the capability is there. The reality is on 63x, you're probably going to be focusing on very specific areas of the slide, but you've still got the capability of getting a large, beautiful image at any magnification. If you're an expert, then yes, you can focus in on the area of the slide you're interested in. You can spend time on the scanner making sure you're only capturing those areas. But if you need the help of a friendly local pathologist, it's much easier to actually review the image once it's been scanned and is sitting on a PC, view it on the monitor, than it is to crowd around the scanner and ask the pathologist, am I actually about to scan the right bit? I spent a lot of time with manual fluorescent microscopes capturing single fields of view, and it was heartbreaking because time and time again, it would be afterwards when the fluorescence was long dead, I realized I hadn't quite got the right bit. So I'm a big fan of this capability to whole slide image. And the reality is that most of the time you would be using 20 or maybe 40x for whole slide imaging. And the 40x is a true 40x resolution because it's using a 40x objective. It also generates whole slide images from individual tiles or fields of view that are captured sequentially as you go across the slide. So it works across the short axis of the slide, moves, goes into position, takes the counter stain, then takes the fluors or the exposures or the Z stacks, moving the dichroic each time. The slide itself never moves. Once the tile is fully captured, then the scanner then moves the slide on to the next tile and does the same thing. And it takes the whole run of tiles and it goes back to one side of the slide again and does the next run of tiles. And then from that, you get this incredibly beautiful seamless image that you can see here on the right. The lines that are showing there are actually lines I drew on to show where the actual tiles are incorporated, and they're overlapping and they're merged together so you get this beautiful seamless tissue to look at. You always get perfect context in all your tissue sections. So generating all these multiplexes is easy, right? Well, it would appear so. but I can promise you managing them can be an even bigger headache. I mentioned that you can get hundreds of gigabytes in an image. Well, I actually have seen 30 gigabyte images being circulated. It was really fun and games trying to get those around the business to different sites. It's put it on an external drive, you can send it through the post, but what happens if that gets lost? It's incredibly easy to lose these images or even delete them by accident if you're not actually using a proper image management system. Sharing images, as I said, you can post an external drive. You certainly can't email something that's bigger than about 20 or 30 megs these days. File sharing platforms, in my experience, have topped out at about 500 gig per single file. So even though I might have a terabyte of space on one of my sharing platforms, I'm actually only limited to 5 gigabyte size files. So instead, if you were to use something like our image management software called Aperio Image eSlide Manager, sorry, I'll say that again, Aperio eSlide Manager, it will manage and share your images faster and store them more securely. And this is my third topic here, the managed part of the talk. The eSlide Manager is really easy to use. All the files are sitting on a single central server, and you use a web-based application to log in and work and access and view the slides. Every login has its own permissions, so some people might be able to view slides, other people might be prevented from viewing certain slides, you certainly can stop people from deleting slides. With its robust built-in security, your valuable research is going to be protected from prying eyes and also, importantly, the delete buttons. Are you a single lab with everyone there every day? Well, Workstation may be the option for you. Basically, you will scan all the slides to and point them at a single PC. Everyone would then walk to that PC and access the image management on that PC and also access a range of image analysis tools to help you interpret your multiplex images. That's a pretty good, neat solution on a small scale. You can scale it up, and instead you can actually go to a server-based solution. If you're a large lab or a core facility, do you have important collaborators in other buildings on campus or even off campus? eSlide Manager, in its server solution, is at its most powerful and flexible. You can hook up all your scanners to the images direct to the server. Each image is ready within seconds of a scan being completed. It's not about waiting for the whole batch to complete. Every single image is treated as an individual file that gets uploaded. You have control over each login and what it can do. So local users can just access it from a PC within the LAN. Remote users can use something like a VPN to get secure access. So you can work not just from home, but also from the other side of the world. If you've got a satellite site that might need its own image management, it's got its own scanner and local storage server, you can do that, and those local users can look at their images, and they can also look at the main site images as necessary, so it's easier to share between the two sites. It's all about permissions. What's of interest is, we do get a little bit of surprise, Aperio also does image analysis. This is the fourth and final topic in my talk. Believe it or not, this is the reason why we actually digitize multiplex slides. I bet you're thinking, huh, right now? Well, people don't do multiplex assays to scan slides. They actually do multiplex to interpret complex biology, ask really difficult questions. Is what immune cells are infiltrating that tumor? How is that going to affect whether that tumor is going to progress or whether we can kill it off with drugs? That's the kind of questions people are asking. They're difficult questions to answer, and that's why multiplex is being developed. Image analysis can actually help you with that interpretation. And more importantly, although you can get a lot of data by looking at the images digitally yourself, it's actually algorithms are able to mine even more data out of that. We call this data mining in ways that you can't possibly do manually by eye. It's absolutely extraordinary the data you can get off this stuff. Our Aperio algorithms have been out and about now for about 15 years, and they're used by more than 300 organizations in 40 plus countries. With more than 300 peer-reviewed publications citing the algorithms, we must be doing something right. So let me show you a bit more about them. This is the research use image analysis menu. 14 algorithms divided into machine learning applications, analysis of proteins in immunofluorescence and immunohistochemistry methods, and molecular analysis, which is actually nucleic acid probes such as RNA and DNA. The numbers on each algorithm actually indicate the numbers of colors the algorithm will support. The U on the left there for Genie and TMA Lab is actually for unlimited. It's hardly surprising that a tool for segmenting and managing TMAs doesn't care about Plex. Actually, neither does Genie. And Genie is our powerful pattern recognition software that can identify your regions of interest. So with Genie, you can make your own robust tissue classifiers that look for your tissue, your particular stains, and direct analysis for the correct areas of your slides. So you can become very expert without having to refer to a pathologist each time. You can have the pathologist help you build a classifier, and then you're actually freeing up their time. So there's no more annotations and no more mistakes, especially in highly heterogeneous slides. Apart from the machine learning tools, Genie and TMA Lab, cellular immunofluorescence is the winner in the Plex stakes. It's the top left in the protein section. It supports 9 colors. Specifically, it supports 7 targets and 2 counter stains. And I'll tell you a bit more about this outstanding algorithm in a minute. It's a perfect example of the revitalization of the Aperio image analysis program that is answering customer needs for multiplex now, and really does let you know that image analysis from Aperio is open for business. What I will point out is our bright field algorithms are all 2 or 3 color. Well, this actually means that it's 1 or 2 plex targets. Do we really care about that? Well, actually, most bright field imaging is still 1 and 2 plex. So we are able to support the vast majority of immunohistochemistry and chromogenic-ish assays that are out there at the moment. But watch this space, there will be more to come. So here it is. Let's go through the Aperio cellular immunofluorescence algorithm. It's a bit of a mouthful, but it's worth it. We designed it to mimic how you'd work down a microscope. What do I mean by that? If you look on the right there, you can see the different tabs, how we've ordered the parameters. So you basically can go through, what am I looking at? Would I say that's a cell? Would I say that's positive staining? How would I score that cell? Is it type X or type Y? What are my final totals? If you take that image on the top right, you can end up with a markup that's looking like that on the top bottom right. To model the cells accurately, we've industry-leading double counter stain cell modeling. There's the standard nuclear counter stain. You can use the DAPI, or you could even use that red stain that's only marking up some nuclei. You can use any stain you want for your nuclear marker. You've then got a choice of either painting on the cytoplasm and the membrane to set depth, or you can actually use another counter stain or another dye, in this case the green stain in that slide, to model the cytoplasm or the membrane just as accurately as you are the nucleus. So you're modeling beyond the nuclear boundary with confidence. Why does this matter? If you just paint on cytoplasm with a set depth around the nucleus, not really giving the real shape of the cell, you could get suboptimal results and really get confused. You can see here that I've actually used the green fluid to model the cytoplasm, so the cells are more cell-shaped, and the cytoplasm only exists where there's green stain, and I think that's pretty neat. Multiplex staining patterns can be complex. I show you here 2 targets, a red and a green, and a DAPI counter stain. But how do you keep 7 targets straight in your head? They'll all have different points of threshold between what's positive and negative staining. They might be in 1 or more different cell compartments. It's not too bad to do 7 stains on this 2 dozen cells that are in this image here. But what if you've got to do a whole slide? That could be 500,000 cells or more. You keep that straight in your head for 7 stains over 500,000 cells. Good luck, because you're a better person than me. There's no way I could try it. And then you've got to remember, you've not just got the 500,000 cells on this slide. What about all the different cells on the other slides that come off your batch of 30 from the Bond Direct stainer? It's much easier to characterize a cell population with automatic classification from this algorithm. If you're doing or intending to do multiplex assays, it makes sense to use Aperio image analysis to generate those numbers for you. So what's the cell here and there to the overall result? Well, actually, every cell does count. You've gone through all that trouble of staining and imaging all those cells, so you should consider them all when interpreting your multiplex assay. Just look at these three cells here, A, B, and C. Each is a different size, colour, and shape. Totals and averages really are not going to tell you the whole story. The cell data would actually give you a much bigger picture. It's complex, but it could be critical to the quality of your final results. Don't disregard it. That's all four topics I've covered: stain, scan, manage, and analyze. I just want to go through how it all hangs together in a workflow in the Aperio zone. So when slides come out of the wet lab, they're in the Aperio system. You've got a new assay. You make a new versus scan template in seconds, literally just takes seconds. You can cheat and copy an existing one. You then set your exposures, your Z-stacks for the fluids, and go scan. You've got the confidence to know that that slide, when it's finished, the image will automatically upload to your slide management system, and you can see it as soon as it's available. You can have a look at it on the viewer and do what you want with it. If you want to actually use image analysis to help you interpret the image, you actually need to tune an algorithm. In this case, a cellular immunofluorescence algorithm, because it's a multiplex assay. You open it up in the viewer, and we call this tuning the algorithm. You set it up to detect your targets and counter stains, to set your scoring classes, and then you save it as something we call a macro. And that's just a file. It's just a version of the algorithm specific to that particular assay. You then annotate your slides so that you say, these are the areas of the slide I want to analyze. You then tell the system to run the analysis, and you can sit back and wait for the results. You can watch it go it live, or you can walk away and view it when it's ready. The point about saving that macro is you can actually go in and edit it time and time again. You can tweak it until you're happy that it's right. So you're not just setting up the staining and the scanning. You can actually get the analysis interpretation just how you want it. You've got full control then. Once you're happy with it, you actually don't have to tune anymore. You scan your slides, the upload to image management. You then bypass tuning. You just annotate your slides, tell it to batch analyze, and that's literally where you select your slides, you select your macro, you tell your slide manager, go analyze these layers, and it will do that. It will spit out results that you can view where and when you're ready to view. You can have complete confidence within that your multiplex assay will actually continue and complete in the Aperio zone. So let's look at the cellular immunofluorescence algorithm in action. I'm just going to take a really simple 3-color image. We've got tons and tons of beautiful multicolor images, but showing you how the algorithm works, it's actually more powerful just showing you with just a couple of targets. Here, red staining is actually spectrum orange, but we've pseudocolored it red for good contrast. It's the estrogen receptor. Green is cytokeratin, blue is the DAPI counter stain. Pretty standard stuff. I can model this to determine what cells have what stain. So if I just go back a bit, you can see that we've got green in the cytoplasm, we've got red in some nuclei. We don't have all cells with red, we don't have all cells with green. So I want to quantitate what cells are red, what are green, what are both, what are none, what are maybe a few combinations in between, ratios of staining. These are the results that you'll get overall, but I'll go through the per cell in a minute. From this area here, I've got 2,207 cells counted. I can see a lot of data from the overall results. I can see the name of dye 1 is ER, the name of dye 2 is cytokeratin A. I can see how many cells have ER in their cytoplasm and how many cells have keratin in the cytoplasm. Too many cytos. What if I didn't know where the biomarker was in the cell? Or what if it was in several different compartments of the cell? That doesn't matter. With this algorithm, I can actually set any of my 7 test dyes to sit in the nucleus, the cytoplasm, the membrane. I can choose a single compartment or I can choose multiple compartments for each one of those. So theoretically, I could be actually looking at 21 different combinations or locations of my markers. That's 7 times 3 compartments. An extraordinary level of detail we can get with this algorithm. Do you want to know how much of a nucleus has ER staining on average? Well, it's there too, and it's actually telling me it's 22%. So this ER staining is not all over the nucleus in every cell. It's very patchy, and you can actually start to see, infer different things from that. The numbers are also of the scoring classes are present there. I've color-coded these to make it easier to see. So you can see class 1, class 2, class 3, class 4, and class 5. You can see the range of the numbers of cells I've got in each of those classes. Color-coded these to match the markup that you get on the mask, just to make it easy. You can see that most cells actually fall into class 5, which is actually ER negative, CK8 negative. That's because I set the threshold to what I would consider positive staining quite high for both of those. If I set those lower, I would have had fewer cells in my negative class and more cells in those combinations of positive classes there. Note that I used cytokeratin to model the cytoplasm accurately, but I also analyzed it. So using something as a counter stain did not exclude it from analysis. And I think that's really useful. What about if I wanted to just know how strong is the intensity of the eRNA cells? This is like the standard immunohistochemistry assay that people would do in a breast panel. So you'd want to know 1 + 2 + 3 + staining for ER and tumor cells. Well, I can identify the tumor cells because they would be green. They would have that CK8 staining. I can then set 4 classes. There's one that's negative for ER, another one that's low, 1+, medium, 2+, strong, 3+ for ER positive nuclei. I can then save the macro, run the analysis. The result is still got 2,207 cells, and the result is it's 2+ line, because actually you don't have enough cells in the 3+ class, the high intensity class, to actually constitute 3+. You have to have at least 10% of cells there. You actually reach the 10% when you're in the 2+ class, so we actually can say this is a 2+ result. Do you want it all? Well, you can have combinations of biomarkers and their intensities and the area in which they occupy the compartment that you're assaying for all 7-plex. You've got huge amounts of data you can generate from this right there on your PC, and all you've had to do is set up the macro once. So this is per cell output. It's actually a very, very long table here. It's a grid underneath the overall results. The overall results, you're only seeing the date, start and end time, so you can see the audit trail there. But here in the results, I'm actually displaying 6 cells here. Each row represents a cell, and you can see that you've got data like the presence or absence in a class, the area of nucleus, the area of DAPI counterstaining. You can actually find out an awful lot of data for each cell from this. And if I was to page down, you would see that I would actually be able to view all 2,207 cells. I can get the area and shape of each cell. I can get data on intensity, concentration, area of staining for every fluor in every cell. And there's so much more. Because people want to do stats on these cells, we can do a one-click export to make a CSV file that can go into any third-party spreadsheet application. And I often do that. And you can start to filter and crunch down the numbers. You can filter here. You can see that I've got a filter on the presence in the orange class, presence in class orange 3+. You can see that I'm only showing the ones there, and they're the ones that are present in the class. When you're doing stats on these cells, I've got 2,000 cells here. What if I was to do stats on 500,000 cells? That's pretty significant. But what if you've got to score 500,000 cells on each of those 30 stain slides that came off the bundle at the beginning of the talk? That's 15 million cells. That's a lot of stats for you to play with. You don't just have to rely on the stats. You can review your cells individually, as you can see here. I'm able to filter on that class, presence in class orange 3+, and I've actually highlighted that cell that's called 1323. You can see here it's indicated on the markup with a red box around, and I know at a glance it's a 3+, because I can see there's a 1 in the 3+ column in the grid, and also it's dark red. And I set up this assay a little bit prosaically that my blue is going to be negative staining, my yellow was going to be 1+, my tan was going to be 2+, and my dark red was going to be a 3+. Software will just help you sort through. So I can click on each of the cells in that grid or multiple cells at a time, and they'll all highlight in the markup. That's great looking at the markup. But actually, I can hide the mask and look at the cell itself underneath, and I can see that that's an intensely stained ER cell, and I can actually look at the other cells surrounding. And if I was to go backwards and forwards on this slide a few times, you can see why some of the cells are blue, they're negative. You've got 1 1+ cell, and the rest are 2+. Okay? So that's actually how you deep dive into every cell. So I want to end with a final thought. Ask yourself, what would you aim to count per slide manually? Back in the day, I'd be doing 100, 500 cells, 1,000 if I really was pushed. Hated doing it. But could I do 1 million? Not happily, not on one slide, not without losing all my marbles. And I've got so few of them left. What about those other slides in your bond batch? I was speculating there could be 15 million cells at least on those slides to look at. It's all adding up, and crucially, it's backing up rapidly without the aid of automation to handle that kind of data. So, on that final note, I'm going to thank you for your attention today, and we'll see if there are any questions. Thank you.
Thank you, Dr. Snyder and Dr. Loupart, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Dr. Snyder, this first question is for you. How many markers can you detect simultaneously?
That's a great question. We routinely detect 4 markers simultaneously using 4 different peptide-hapten, anti-hapten pairs, but we are continuously expanding our library of peptides and can detect 4, 5, or 6 markers at the same time if needed. Essentially, the only limitation will ultimately be the number of fluorophores you're able to detect simultaneously, and that you need to distinguish one from the other.
Thank you. Dr. Loupart, let's have you answer this one. Do you need different kinds of scanners for FISH and immunofluorescence?
Actually, the answer is yes and no. The 2 techniques do benefit from different resolution and scanning methods. FISH really does like the 63 and the 40x, does like to use oil. Immunofluorescence you can often get away with. I've seen 10x out there. I've seen 20x a lot. 40x is becoming quite popular for immunofluorescence. I actually am now seeing some 63x for immunofluorescence. If you've got a standard line-based scanner that you're using for immunofluorescence and you want to do FISH, the answer then is yes, you do need different kinds of scanner. Line scanners usually use a 20x or use a doubler lens to generate a 40x image. And these won't have sufficient resolution to resolve or separate those 2 closely located FISH spot signals, especially if you're doing a break-apart fusion assay. What tends to happen with line scanners, not all, but the one I'm thinking of in particular that we do have in our portfolio, is that it captures each fluor in a strip from side to side on the slide, and then it aligns the strips afterwards. Well, there's a real alignment issue there. So how could you be sure that you've got the confidence your great fusion assays are as clean as they could be? FISH really does benefit if you've got a tile-based scanner, where all the fluors, Z-stacks, are captured in one before the slide moves on to the next tile. So it's much more likely you then just have to make sure the tiles are aligning, but you can look at the individual cells in each of those tiles and know they're properly aligned. All the signals are aligned. If you've got Versa, the answer is no, you do need different kinds of scanner. You've got a 2-for-1 or best of both worlds sitting on your bench. You've got a wide range of objectives. All the oil and Z stacking. It's even got this fabulous little FISH enhancement setting that I never went into today. It really does clean up your FISH images because it's actually helping you use the DAPI mask to make sure you're getting the right FISH signals within the nuclei compared to potential artifact staining outside the nuclei. Really neat little system. IF and fish can be done together on the same instrument. And the really brilliant thing, as I said before, you can actually run them in the same batch using different scan templates. So with Versa, you don't need a different kind of scanner, but some other scanners can be limited, and you're usually settling for a fish or a fluorescent scanner. There you go. Thank you.
Okay, Dr. Snyder, back to you. Are you able to detect mouse antibodies on mouse tissue using UltraPlex?
Yes, one advantage of the UltraPlex technology is that it separately barcodes each primary antibody with a different hapten and detects using anti-hapten antibodies instead of anti-species specific secondaries. So it makes no difference to us either the species of the tissue or the species of the primary antibody. This means that the technology has a very wide applicability in a whole variety of models.
Dr. Snyder, let's piggyback on that last question. Can you tell us on what other tissues has UltraPlex been used?
So UltraPlex can be used on both FFPE, formalin-6, paraffin embedded tissues, and also frozen tissue sections. We have stained a wide variety of tumor types using UltraPlex panels. Off the top of my head, these range from multiple subtypes of breast cancer, head and neck squamous cell carcinoma, prostate cancer, lung cancer, squamous cell carcinoma of the cervix, gallbladder adenocarcinoma, thyroid gland, Pillory carcinoma. I think we just looked at some of those images just a few minutes ago. Melanoma and pancreatic cancer. We've also stained renal tissue with glomerulonephritis, and of course, we always routinely stain normal human tissue, such as tonsil, and development of all of these panels. But there's obviously no reason why this technology could not be applied to many different tissues and disease states. And we have indeed stained other tissues also.
Coming in for Dr. Loupart. Dr. Loupart, how do you decide on what fluors to use for multiplex?
Well, Bond, Versa, and cellular immunofluorescence algorithm can all manage many different combinations of fluors. So you can just continue to work with your favorites that you already got in the lab. You don't have to spend time validating a new stain or a new detection methodology, which can be expensive, and it can really drain resources as you try to replace your existing methods that are already working. A careful choice of narrowband dichroic filters on the scanner will actually reduce overlap or bleed through of fluors. And if you look at that range that I talked about earlier, from DAPI at 350 to size 7 at 750, it's possible to have 4, 5, 6, or maybe even 7 separate fluors. So if DAPI and size 7 are actually 2 of them, you could easily drop in the likes of aqua, spectrum green, maybe a spectrum gold, some sort of spectrum red or Texas red, getting in size 7, and also dropping in size 5 between the red and the size 7. It is possible to get up there. We need to do the tests, and we need to check that it works for your kind of assay, but I believe it's possible. The software then will just actually take all the images that come off the Versa. Each is retained as its own grayscale, and it will then just interpret each scale and give you a final full color result. It's simple, flexible, and it keeps you in control of your assay at all times.
Loupart, let's stay on this topic for the next question. How easy is it to manually stain multiplex?
Okay, so manual staining is pretty laborious. It takes up a lot of a technician's valuable time, all those steps, all those washers. Even technologies such as Cell IDX's UltraPlex, they will reduce many headaches, but you're still looking at a lot of steps potentially. There's a lot of variation. You just need to get distracted by a phone call or something, and your timings could be off when you're doing this manually. The BondRx automatic stainer is a setup and walk-away system that will save time and provide consistency whatever the stain technology in use. So you set it up to go, and bar Armageddon, that thing will finish your staining for you, and it will be the same as the previous batch, time and again. The user still needs to ensure the staining is working correctly in the first place. You have to set it up and check that it's as good as your manual. You've got to make sure your antibodies are well characterized and they're actually locating where they're supposed to. There is a lot out there in the literature at the moment that, no two antibodies from different vendors might actually be hitting the correct target because they've all been generated in different ways. You've got to be very clear on what you've actually created, especially if it's a homemade antibody. It's also important that the antibodies in the detection system are titrated correctly. So you're reducing the chance of cross-reaction or even blocking as targets. And these are just a couple of examples. Working with BondRx takes some of the labor away.
And while we're talking about staining, Dr. Snyder, can you share with us how long the staining process takes?
Basically, the staining process takes 2 hours and 2 steps. All of our hapten-lated primaries and anti-hapten secondaries are applied as cocktails. That's the really nice thing about this technology. So regardless of whether we perform a staining for 3, 4, or 5 markers, the process is the same. It's a 1-hour staining with the primary cocktail, wash, and then a 1-hour staining with a secondary cocktail. There's only one antigen retrieval step prior to this, so it saves a great deal of time compared to other techniques which use multiple different antigen retrieval steps.
Dr. Snyder, one more question for you. Now, we see that other technologies use multiple antigen retrieval steps, but yours only uses one. Why is this?
Well, firstly, we screen our antibodies on FFPE tissue under different antigen retrieval conditions, and then intentionally select a condition which works well for all the antibodies in the panel. It was very important to us that this technology be as simple, robust, and easily adoptable as possible. Second, we have only one antigen retrieval step because we have simultaneous binding and detection of all markers. So unlike other technologies, there's no need for multiple rounds of stripping and staining or optimization of different individual antibody pairs in terms of the detection sequence, the order in which those antibody pairs have applied to the tissue.
Thank you, Dr. Snyder. We have time for 2 more questions. Dr. Loupart, let's have you answer these last 2. How does the SW manage to segment cells accurately?
Multiplex algorithms, that's cellular immunofluorescence, and actually also the 2 FISH algorithms that I didn't really talk about, have more than 15 parameters used to identify and segment the counter-stain fluorescent nuclei. Is it overkill? Probably not. They do use traditional parameters initially, such as merging, smoothing, size filtering, but then we've also incorporated things like noise reduction and machine learning to handle those crowded and overlapping nuclei more effectively. It can be quite tricky, especially in fish, where the cells are subjected to a huge denaturation shock. To actually see the individual nuclei, they tend to look a little bit different than you do in immunofluorescence DAPI staining. So our algorithms are designed to actually combat that kind of issue. The live tuning window that we have gives real-time feedback when you're tuning to see how effective segmentation is. As you change a parameter, you can then see within a second or so what that change is to in the live window. In after analysis, if you really don't want some of the nuclei, it's marked up, some of the cells, you can actually edit them. You can delete them out. You can do that at the point in the grid, the per cell grid, and then export the results, or you can actually do it in third-party spreadsheet later if you don't like it. There's a lot of control there at each step.
And let's end with this great question. Is automatic analysis really faster than in-person scoring?
Aha. Man or woman versus machine, the old question. Well, to be perfectly honest, for 1 or 2 plex assays on a few slides, it's probably not going to be faster than the machine. But when it comes to multiplex, the limitations soon mount up, and digital really starts to win out for speed, accuracy, and reproducibility. It's all about how many markers can you keep straight in your head? What do they all mean? The cutoffs for each marker can vary, and overlapping cells can make manual scoring incredibly difficult to remain consistent. The algorithm will handle that. And the slides, when they mount up in number, the user does get tired, but the software really doesn't. So you can generate counts of 50, 100, 200 cells quickly manually, but you're not going to get into thousands unless you're using software to help you.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Just a reminder, any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. This webcast can be viewed on demand through October 2018. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.